that might facilitate other kinds of research. And just a couple of quick CCE announcements. You may recall at the last CCE meeting we talked about whether to submit a bolus of papers related to the rapid award we received from NSF in relation to the 2015-16 El Nino. I'm pleased to say that 5 manuscripts have been submitted to Deep Sea Research 1, and they are in review right now. And that's a nice accomplishment. Also, for people's, for your interest, there's the LTER All Scientists meeting that happens only every 3 years. This year it will be in Asilomar, California, rather than up in the Rocky Mountains of Colorado. So in Asilomar from the 30th of September through the 4th of October. And that's open to all. Okay, so today there are a number of people in the extended CCE community who are either in the room or remote. Maybe I'll ask people who are remote just to say hello and give us your name so people know who's listening in. So Manu, could you start?
Sure. I'm Emanuele Di Lorenzo. I go by Manu, and I'm at Georgia Tech on the East Coast.
Hi, this is Chris Edwards. I'm up at UC Santa Cruz.
Hi, I'm Fanny Chemia, and I'm in France right now.
So, sorry, Fanny, where are you right now? Where in France?
Brest, the rainy part.
Okay. Thank you, Fanny.
This is Matt Long and Jessica Liu at NCAR in Boulder, Colorado.
Great, glad you could join us. And do we have someone else?
Mike Jaycox.
Mike Jaycox and Stephen Bograd at the NOAA office in Monterey.
Good, glad you guys could join us. And Stukel, are you there? Okay. We had a little crackle there. We'll assume that was the Stukel crackle, and then there's no, Chris already identified himself.
Oh, I'm sorry.
Okay, so I was originally planning that we might go large scale to small scale, but I think it's going to be broken up a little bit because Peter has a class to teach, etc. So it's going to be a sort of mixed mode. But we're going to start with Manu and some of the things that he's been thinking about. And by the way, let me remind speakers and other participants, part of the idea is for us to become aware of what's going on and perhaps to seed new collaborations. So Manu.
Okay, thank you, Mark. So I will be talking about 2 projects that are ongoing here at Georgia Tech, some in collaboration actually with Chris Edwards at UC Santa Cruz and also with Art there at Scripps. The first one is about ENSEMBLE eddy-resolving modeling of the North Pacific Ocean circulation. And here, one of the goals is really a physical basis for decadal predictability. And this is an NSF project that is currently in the 3rd year. The goal of this project is twofold. The first one is really to understand the generation and propagation dynamics of subsurface physical, biological, chemical anomalies in observation and eddy-resolving models. And the second one is to see whether we can exploit these subsurface dynamics to make decadal predictions in the upwelling systems of the Northeast Pacific. In this particular case, the Gulf of Alaska and California Current. And before I move any forward, let me just say one thing that I forgot, that this work is mostly from a PhD student that graduated last December, Mercedes Maria Pozzo-Buil, who is currently working with Mike Jaycox up at Monterey. So that's within the family, I guess. Very good. With that said, the general conceptual model is very simple. This is a schematic of the Kuroshio extension region, the North Pacific Current, California Current, and Gulf of Alaska. And so the real idea is that in the subsurface, you have anomalies that propagate down the stream. And as they reach the upwelling systems and they come, essentially at the surface, those anomalies essentially carry inherent predictability on timescales that is actually up to maybe between 5 to 10 years if you monitor the anomaly in the North Pacific Current. In previous work that was discussed probably in some of these talks, Maria Pozzobuil showed that, in fact, you can predict part of the low-frequency variability of sea surface temperature in the Gulf of Alaska through this mechanism. And more recently this year, she published a paper about how oxygen anomalies are also predictable potentially in the CCS upwelling system, although the predictability needs to be further kind of measured. With that said, as I said, this is a potential predictability of up to 10 years because of the slow propagation. There are different generation mechanisms of these anomalies in the subsurface. The first mechanism is really just a simple propagation. The anomalies get propagated in the KOE and then are essentially propagated along the mean circulation all the way into these two upwelling systems. And there's some evidence from modeling studies that I'm not going to, kind of touch on right now. But there are also observational evidence from Argo, and this is a plot from a study by Koketsu showing the propagation of these Argo subsurface salinity anomalies on an isopycnol, which is essentially a density layer in the subsurface coming from the Kuroshiwasho region. Now, of course, the reason why subsurface salinity anomalies on isopycnols are important is because on isopycnols, temperature and salinity are compensated, so a salinity anomaly has essentially an equivalent part in temperature, but also a lot of biogeochemical tracers like oxygen and other nutrients have a similar behavior. That is, they're really controlled by the subsurface advection dynamics. And so in some way, the salinity is really a proxy for a whole bunch of other biogeochemical tracers in the subsurface as they reach the upwelling. Now, one of the problem of these mechanisms, of course, is unclear whether these anomalies really propagate coherently across the entire gyre, and this is something that we are exploring. Another important mechanism to point out in the subsurface is that these subsurface anomalies, as they propagate along the gyre, their low frequency content, or if you like, their decadal energy in the power spectrum gets amplified. And the simple idea is that you start with an anomaly in temperature salinity on an isopycnol as it propagates along the current, and these small fluctuations in the eddy field or other anomalous current can slowly amplify essentially through a reddening. This is a famous red noise model, can actually amplify the energy of these low frequency anomalies. And there's some studies that have been done by Kilpatrick, Schneider, and myself showing, in fact, this amplification process taking place in the subsurface here in the California current. So these are two mechanisms that are together essentially energizing this low frequency energy of the subsurface tracers, and potentially once they're in the North Pacific current region here, they can actually have inherent predictability. Now, one of the questions, of course, is from observations. We cannot establish how robust are these propagation mechanisms. Do they propagate all the way coherently from the KOE? These are questions that we don't know yet. And what are the mechanisms for generation propagation of these subsurface anomalies? In other words, what is the relative importance of the first mechanism, that is just a pure, simple advection by the mean flow, versus the mechanism two, the generation of the anomalies locally as the anomalies are propagating? So to this end, that's where the ENSEMBLE modeling, ocean modeling, comes into play. And in this particular case, we use the regional ocean modeling system, the ROMS. And to do that, we essentially performed a whole bunch of simulation, which I will shortly give you the essence of it. But essentially, we have a very long control of about 1,200 years that we use to spin up the ROMS model at a resolution of 23 kilometers. After that, we use the final state of that simulation to initialize a 12-kilometer ROM simulation. This is the grid of the North Pacific, which is then run for another, 153 plus 300 years, so about 400 years. And then after we do that, then we start essentially a set of 6 ENSEMBLE hindcasts from 1950 to 2015 to essentially have several realization of the eddy fields and subsurface dynamics of the North Pacific. The idea being that by having several realization of the model, we can separate very cleanly the part of variability that is deterministic, meaning it's responding to the forcing, versus the part of variability that is intrinsic. And this is really important because in the subsurface, it's unclear how much of those anomalies are actually deterministically forced by subduction processes at the surface versus, internally driven by the eddy field. So that's really the question. And this is just a very simple result just to show you how the model does. So what you're seeing in this plot, it's essentially the average correlation that exists between any of the ROMS ENSEMBLE and a reanalysis product put out by ECMWF, the Aura S3, and this is an isopycnol 27. So this is like this has all the high frequencies, so it has monthly timescales, and so you can see that there's a region of very high correlation between the ROMS ensemble and the reanalysis product, and then there's a region of less correlation. What's interesting is if we do the same plot, but we do it across the ROMS ensemble, so we don't have the observations, we find a similar kind of spatial pattern. I'm sorry, my children in the house, and so this gives us some confidence. And in fact, these dynamics are, the model is capturing some true dynamics in terms of what is deterministic, essentially this blue region here, versus what is a more intrinsic or internal to the model. And so the idea now is to really diagnose what is the source, what are the dynamics that give deterministic variance in this region versus more intrinsic variance in this region. So that's what we're working on. And this is work in progress. This is an example of a time series of the ROMS salinity, on a particular region where the deterministic variance is very high, compared to the Aura, which is a reanalysis product, showing that it captures this low frequency phasing of the signal, which is encouraging. But it also tells us a little bit that part of the signal is locally generated. It's not just coming from the KOE. Now, why is salinity important? In a previous paper by Pietri, she showed that the salinity anomaly on the isopycnol tracks really well subsurface oxygen anomalies from different sources. This is the coffee oxygen, Newport oxygen, and then a reanalysis of the California oxygen by Deutsch. They're all plotted together, showing this kind of low frequency phasing. So I will skip this part of the talk showing the model propagation, because I want to use my time more wisely. But essentially, you can see the anomalies propagating backwards in time all the way into the KOE. And I'm going to skip this. And what you're seeing here on this particular plot is just a Hahn-Muller diagram showing the propagation along the stream, along one of the circulation stream functions here. Okay, with that said, I want to move forward to another project that we're currently working. This is in collaboration with Art Miller, Chris Edwards, and a whole bunch of other people on the observational side, like in Falk Pedersen's group. And what we're doing here is we're actually using some of the large-scale outputs of the ROMS model to essentially do a high-resolution modeling of the inner shelf circulation. And this is an example of going from a 600-meter grid here. This is the Santa Barbara Channel, all the way down to scales that are essentially, this is a cape here, in particular a 22-meter scale. So we cover a wide range of scales, and I'm bringing this up because I'm thinking that for some of the process modeling studies, this could be handy. But there's also another interesting thing. What we did last fall, we used this modeling framework to produce real-time daily forecasts of the circulation at 200 meters. For a field study that ONR was funding through one of their DRIs. And I just want to show briefly, if this works, our daily forecasts. And this is, again, work in collaboration with Chris Edwards, who provided the large scale. And this is, for example, one of the capes Point Sala, and you can see there's features and stuff. And this is an example of a daily forecast for the sea surface temperature and surface currents. And this was actually quite an undertaking because we had to synchronize between Santa Cruz, Scripps, and Georgia Tech and get all these model runs done on a daily basis. And some poor PhD students was working until probably 3 a.m. in the morning. But it turned out that these forecasts seem very promising. So let me just show you one example of I wouldn't call it validation, but this is, unfortunately, my computer is very slow. So this is an example that just came in a while ago, where in this red color you have an NDBC buoy, and in blue you have essentially the GT forecast model. And you can see that overall the model actually is not that bad. And we are planning, of course, now that all the field data is in, we're going to evaluate a little bit more the scale of the model. But in principle, this could be a model that could be used, for some of the process studies, I think. So going back, this is really the end of my summary. We have two main activities ongoing: ENSEMBLE EDDY-RESOLVING modeling of the North Pacific. The idea really is understanding the decadal predictability of chemical, biological, and physical tracers. And then the second part is this high-resolution modeling of coastal flows up to a scale of 22 meters, which I doubt we need for the LTR, but perhaps the 200 meter, and the daily forecast could be something that one could use in cruises down the line. So here is just a list of the model activities. Sorry, these are the list of the research foci of LTR, and I think that this modeling could definitely contribute to a few of these items. One is this kind of modeling of the localized food web changes in response to changes in ocean temperature, and of course we can definitely provide interface with these transport studies, both along shore and cross shore. And this is all I have. Thank you.
Thank you,
I
Mark.
took 12 minutes exactly.
Good. I'm sure there are questions and comments.
I have a question from this is Peter.
Hi, Peter.
Hi. When you were showing the North Pacific the patterns along the North Pacific current and then the bifurcation, and you said that the low frequency energy increased along the North Pacific current. Did it actually increase, or did the high frequency stuff disappear?
Well, so the idea is that what happens is the high frequency variability gets integrated into the low frequency. So this is the famous red noise model. So if you remember, there's the simple, first order regressive model, and that's one thing. But in the subsurface, what happens is these tracers are essentially integrating the anomalies of the geostrophic flow in the subsurface. Now, the geostrophic flow at the large scale is also already red because it has integrated the atmospheric noise. And so what you get in the subsurface, you get these double integration effects, a little bit like the paper that we had with Mark on these double integration kind of model. And it happens in the subsurface for tracers on isopycnals. And so that's how you kind of filter out, or integrate, if you like, all that high frequency, and you kind of filter that out and remain with that low frequency. I hope it makes sense.
It does, but I'm thinking more about the absolute magnitude, and I would still think going through a double integration like that the magnitude of the low frequency oscillations, the amplitude would decrease when it goes through that filter.
If you write the equation for the variance of the tracer, you find that actually the low frequency energy of the tracer, the low frequency energy of the forcing, in this case of the geostrophic currents, gets amplified.
Okay, so the variance is
because it goes. Yeah, the variance. Is the variance conserved? It must be. Yeah. Now you get me here on a math question, but yeah. But it goes like the variance of the tracer will go like the variance of the forcing divided by 1 plus minus something. So then essentially it increases the variance of the forcing. So actually it's not conserved. And so Bruce can correct me.
I'm not getting into it.
He can, but he's too polite to do it publicly. But both of your processes are highly relevant to CCE. I would say that, as you've already said 2 or 3 times, that the process number 2, the focus on the high-resolution modeling of the coastal intercoast process, if you can extend that to a domain of, let's say, 150 to 200 kilometers offshore, that's the scale over which we're analyzing the upwelling filaments, the cross-shore fluxes associated with upwelling filaments, fluxes of mass, nutrients, carbon, and organisms, and the short-term time evolution of those organisms as the filaments move from east to west. So this, is it a simple matter to scale up a bit?
So this model, we've had quite an experience now with Chris Edward at UC Santa Cruz. So Chris runs the 1 kilometer model, or also the 600 meters. I don't remember, Chris. You can correct me. But essentially, he runs almost the entire coast of Southern California. And then we downscale that at 600 and 200 meters and so forth. So for example, if the filaments that you're looking at, I presume that a 200 meter scale would be plenty. And that's, sorry, go ahead.
Yes.
Yeah, and running the 200 meter grid is actually quite straightforward, I would say. I wouldn't say it's simple, but it's straightforward, and we could potentially run experiments of the 200 meters and potentially even configure it on one of the servers for some LTR students to play with the configuration and add some biology or other things. But that's actually quite doable.
Yeah, I'll just add in there. So we run 3-kilometer CCS wide model, and then scale that down to 1 kilometer, and then we pass that along to Manu for the 600-meter run. Anyway, Mark, in answer to your question, I wouldn't think about scaling up what was done here, but rather just stopping the scaling down at an appropriate scale.
Right, okay.
Correct.
Very good.
Chris, you're so precise. Perfect. Good.
Okay, I think we're going to move on to Bruce, and I think you work on multiple scales as well.
Right, yeah. Now, not either as large or as small as mine.
I think James has a clicker if you want it.
Okay.
You may have to enable content up there, the yellow band.
Maybe he can just advance it, right? It's not too crazy. Okay. Okay, thank you. I'm going to talk about just kind of a short laundry list of models that come from a bunch of different projects that Ganesh Gopalakrishnan, who's here, Matt Maslov, Ariane Verde have contributed to, and the more or less results are available on a website that Matt runs, and also on one that's affiliated with ECHO. And so here's the not quite Manu in both directions. One is we've run a 1/16th degree 72-layer 4-year state estimate for the California current. Matt Maslov did that. So that means 4 years. I'll explain what that means, but the long time lets you do things like correct the forcing and even look at geoids. And he's added, Matt and Ariane have added a biogeochemical model, and we've also put in tides. So we have those potentially. Again, this is in terms of advertising wares here. And then Ganesh has done 3-month state estimates, compromising a little bit from the long one to a kind of an intermediate one for 2007 to 2017. And some people are using those, including perhaps Laura Lilly, who might use it for context in something she has. And then we also have 1 kilometer, and could be smaller, but we're using just 1 kilometer now to save time, a one-way nested interdomain. It has tides. Right now it's forward only, but it's supposed to be used for a state estimate to analyze a small-scale experiment in the Southern California Bight, actually in the Santa Barbara Channel. And the last thing is we have a model, the same model, MIT-GCM, coupled to the weather research and forecast model. So we're also hoping to use that in the future to do some analysis. It's not as directly applicable, but I wanted to advertise it. So what I mean by state estimate is it's something that Chris Edwards does. A lot of people do it, and we all just do it on slightly different timescales. And what we're doing is we're fitting a model to the observations, and we can do that with a tool called the adjoint, which lets you take the derivative of the thing you want, which is usually a cost function that you want to minimize, and the cost function is usually the difference between the model and the data. So you get a derivative that tells you how to adjust the initial conditions, the boundary conditions, and the forcing. You can adjust all those, and then you have a free forward run of the model. So the only things that are controlling it are the forcing and the initial conditions that you set up. And that means that if you want to do diagnostics and the dynamics, you can do it. And it means that you force the model. It's a test of the model. You force it to match the observations. And so I've already said that. But it means you can't really fit the data completely. You're depending on your model, and it's a way to highlight model error. And I should do this because it's on my computer. And this, I already said this. We have the outer one is 1/16th degree, 72 levels, and we've run both 4 years. I can't show the thing, but the bottom line, 4 years, 2007, 2010, and then we went to 3 months to get closer to near real time and to be able to see eddies better. And the observations we use are along track, not mapped, altimeter, sea surface height, temperature and salinity profiles that we can get, including elephant seals, which are kind of a cute one, geoid constraints, because we separate the time mean and the anomaly, so we are also refining a geoid at the same time we're doing the estimate. Sea surface temperature, and we have used moorings that Uwe Zent has put out. We didn't use Cal-Coffee stations, we withheld them as validation to compare against. And this is just a cartoon of, shows a model domain, shows the sea surface height on a particular day. You can see a low, the scale is minus 25.5 centimeters is anomaly compared to 22.5 centimeters. And so you can see it's low inshore and high offshore, so it's a southern, at the surface anyway, a southeastward going California current with lots of eddies in it, in this case. And then cartoons of the various observations around the outside, including the elephant seals in the upper right. And this is just one result that I like from this. Because it has to be consistent for 4 years, we get a constraint on the forcing. We correct the forcing. So we're using atmospheric forcing from a model. But because the gliders, we have all these observations that you see in white, we can infer that the shortwave down, that means the shortwave light that's warming the surface, is overestimated by the atmospheric model. They don't have enough clouds, enough stratus clouds. And so you need to correct the forcing in order to have it match. And with kind of a 4-year telescope, you can make this kind of an estimate. And we have checked this against calculations that Dan Kane and Sam Michabelis did with a satellite product. But this is as far as it goes. We've never managed to try and publish this, but a lot of models, and I still hear it now, are biased warm when you run it with just the forcing off the shelf. And I think that it's well known that the stratus clouds aren't often reproduced in models, and so this may be one reason for that, is that you have the atmospheric model will make it warmer than it really should be. And so we put tides in the model. This is the red is model output, the blue is gauge. This hasn't been tuned, but it shows you that it has tides and it's reasonable against a tide gauge on Harvest, which is an oil platform off of Santa Barbara. And then Matt and Ariane have put in a biogeochemical model that has a lot of components in it with a notable absence of chlorophyll, which is diagnostic. But they are using this mostly in the Southern Ocean, although they did do it in California, in the California current region, just in a forward run, not assimilated, but in a forward run, they computed, for example, dissolved inorganic carbon, and you can compare the PCO2 that the model produced to moorings in the CCE. This is something that Ariane did. This is just a forward run to check out how things were working. So in this case, the model is the red line, and the black line are observations, are the 5-day averages of the observations. So you can see that it has something to do with reality, even in an open loop like this without being assimilated. But this isn't active right now. They're spending most of their time doing it in the Southern Ocean. And so we're now using these 3-month state estimates that I talked about, which allow us to get closer to real time. And Catherine Zaba, Kasia Zaba, and Dan Rudnick are using them as part of an analysis of their glider data. So I've drawn on this is Catherine's figure. She's drawn on there 3 Cal-Coffee lines where the gliders go. And the red arrows are the glider currents, 0 to 500 meters average over this time period, and the black lines are the model. So you can see that although it's not quantitative, you can see how the model currents match up to the glider currents and how they provide the context in between the glider lines. And that's the reason for using the state estimate, is it's a dynamical interpolation. And the longer you go, kind of the larger scale you can track things. It's like Manu's propagation across the Pacific, but the smoother your result is. And so as the menu of state estimates go, ours is kind of larger scale and smoother than some that are smaller scale and have more fast moving. And this is a second figure from Kasia showing the potential temperature anomaly, Hofmuller diagram. So the plots are distance from shore in kilometers, 0 to 300 going from east to west, and then time going from 2007 to 2018. And the state estimate, Ganesh is rerunning it to make it up. And you can see things that happen, in particular the time of the blob and the El Nino are interesting. This shows a matchup between the two, which you kind of expect because we assimilated the glider data, although we didn't assimilate the 0 to 500 meter. And you can see along the side the large shore average temperature time series, seeing with the black line is ONI, the Oceanic Nino Index. So you can see how the two of them look, how close or far away we are from each other. And the idea is that we supply information in between the glider lines. And we also have a 1 kilometer model going to 500 or 250 meters of the Santa Barbara Channel region. Right now it's nested. It has tides, but it's forward only. And just here's an example. There's a tidal comparison you can't see anything of up there, but it has tides. And below is a snapshot of temperature at 15 meters in the area. And the experiment that we want to analyze is just north of Santa Rosa Island. So we're going to be trying to use this as a tool for analyzing those observations. And it'll also be used for looking at the interaction of internal tides with eddies as part of the surface, the SWOT mission, which is a surface water ocean topography satellite that it's an interferometric SAR, synthetic aperture radar satellite, that will in principle look at very high resolution ocean surface topography, but that means you get not just eddies, but tides, even surface waves, and so we're going to be trying to unscramble all those different things. And that's it. So we have these different models, and from large to small, and state estimates that hopefully can be used as a context. So questions?
Excellent.
I didn't keep time, but
no, you're doing fine.
Okay, move on.
We'll go.
Can I ask a question?
Yes.
Bruce,
Yes,
two questions. I didn't read the resolution of the model, and also how long does it take to perform like a state estimate, say for a month period or something, or how long is our state? I may have missed it at the beginning.
The model that we do the 3 month. Oh, thank you. We do 3 month state estimate is 1/16th degree, so about 7 kilometers, and on it depends where you run it, but on our local cluster, right, it takes about a day to do an estimate for 3 months, a state estimate for 3 months. And
wow.
And something else is that Maslow, they've now, and Ariane, they've decoupled the adjoint so you can run the adjoint at a different resolution than the forward model. And we hope that will speed up the process.
Very cool. Nice work.
Thank you.
Okay, so next up will be Peter.
So thank you, Mark. This is really more a problem looking for a model than models. I have some simple kinematic models in here, but it's a, sorry? Not yet, we'll get to that. Okay, so sub-mesoscale fluorescence and salinity layers at fronts. And Marina Levy, Schaefer-Smith, and I should say this is work with Alain de Vernet, who is a student here. He's now actually a postdoc with Marina Levy and Schaefer-Smith. Marina Schaefer and I just wrote a paper on submesoscales and biology and couplings and interesting things. In the process of doing that, I reanalyzed some data from Alice Pietri and her group that was published in 2013. These are glider transects across the shelf. The top one here we go, depth and distance away from the coast. So the coastline's here. This is actually in the southern hemisphere, so you're looking to the south, I guess. This is the coast. This is contours of salinity, so you can see this. It gets saltier as you go offshore, but you can see these layers of high salinity that seem to be extending down from the surface. And I've contoured in magenta, Jess's favorite color, the 34.9 isohalide, just to sort of highlight that. Now here we have the chlorophyll, log 10 of chlorophyll, and the same magenta contours on this plot. So that's the 34.9 isohalide. And you can see that these layers of high salinity are coincident with layers of high fluorescence. And I plotted these in density coordinates. So this is density and horizontal distance. And the purpose of doing this was to see whether these layers were tilted across isopycnals or not. And so if they were on isopycnal surfaces, so there's a big front here, if they're on isopycnal surfaces, these layers would all be perfectly flat when you plot them this way. But you can see that they're tilted. They're crossing isopycnals in salinity and in fluorescence. And that constrains the sort of dynamics that you might get that can make these. And so I'll talk about that in a minute. But here's some data from the cruise 1208. This is one of the transects. I'll show you where toward the end of the talk. Sorry about this. Hopefully enough of you were there that you remember. But this is Transect 4. So this is across a front, inshore and offshore. So warm water offshore, colder water inshore. The white lines here, this is depth versus horizontal distance. The white lines are isopycnals. And this panel is salinity, and this panel is fluorescence on the right here. And this is Transect 4. There are 4 transects across this front with the MVP, so really closely spaced CTT fluorescence data. And Transect 1 was to the north, and Transect 4 was a few tens of kilometers to the south. And so you can see the same sort of thing. There's high salinity layers here that are separated by low salinity layers, and there are also high fluorescence layers. And these layers are about 5 kilometers or less across. They're fairly small. They fall in the sub-mesoscale range. And if you plot them again with density on the vertical axis instead of depth, you can see that some of these layers might lie along isopycnals, but some of them clearly cross isopycnals. And again, this gives some dynamical constraints on what could be making these. And these, I'm fairly convinced these are real layers. They really exist. They're the same sorts of things that Pietri saw. And my guess is that if we looked at high resolution at most fronts, we'd see these same sorts of features. We just haven't resolved them yet. And in fact, in Stuckel's paper, they looked at these sort of layers, but because of the sampling, I think they all got mushed into one layer when I think it was actually 3 features. And I think that's really significant. I'll show you why in just a sec. So how do they form? Well, this sort of obvious hypothesis might be that there was downwelling that brought this high fluorescence, high salinity water down, and maybe some upwelling here and downwelling there. So you have these tongues of downwelling water that are forcing these layers from the surface where the fluorescence is high and the salinity is high down. The problem with that is to form these layers, you actually have to push water across isopycnals. And water doesn't want to go across isopycnals. It will go along isopycnals quite happily, but going across isopycnals is really hard. So in its most simple form, this is very unlikely to work unless you have some other process going on, like as it gets pushed down, it gets warmed up and keeps going down, but the actual isopycnal changes, which could be happening. But it just seemed to me that it's hard to come up with a mechanism that's going to drive these really small-scale circulation structures that would be persistent enough to drive these sorts of layers. And there are sub-mesoscale processes that can do that, but they typically don't have quite as many branches as this. And I thought this just didn't seem like a very Occam's razor-y sort of hypothesis to me, and I'm skeptical of it. Also, there's one observation by Andrei Sherbina suggesting that this sort of velocity structure might work, but he only had one layer, and it was actually a larger scale velocity gradient. I'm slightly skeptical. Okay, and also that was associated with a bend in the front. This front was pretty straight. So another hypothesis that might seem more complex, I think it's actually more simple because it's kind of kinematic. That is, it doesn't require any fancy dynamics, is that we have tilting of existing patches. And so these layers you can see are sort of angled. And my hypothesis is that these layers became angled and that before they were just blobby patches that might have subtended several isopycnals, that when they came into the front, they got tilted. And so it kind of looks like subduction, but it's not. And so let me explain what I mean. So picture a system, here's depth, and this is our front. So the colors in the background, think of them like isopycnals. So we've got this front, and then we've got these tracers. Let's say it's high fluorescence, low fluorescence, high fluorescence, low, or something like that, that are embedded in the water. They're just tracers of the water, and for some reason they stopped at the bottom here, but they're sort of independent of the dynamics that form this. And then we have some sort of circulation pattern that's trying to kind of flatten the front. So the front is somewhat unstable because you've got light water up near the surface, heavy water up near the surface over here, and it doesn't want to be there. The front's going to try and lay itself down. And so this kind of cross-frontal circulation would be expected, and there's lots of models that show that. This is the same thing plotted just in density coordinates, just showing if you had this sort of circulation pattern, what it would look like in density coordinates. So if we run this forward, he said hopefully. Come on, you can do it. Well, that was good. Okay, try again. James, it's not working.
PC to Mac.
Yeah, this is running forward. Yes. Okay, so as the front's relaxing, these layers, can we do that a bunch more times? Yeah. These layers get tilted, both in real space and in density space, as the front sort of settles down. And so this is kind of what I think might be going on. So let's go to and this circulation would be called an ageostrophic secondary circulation. So let's go to the next movie just to get it over with. So what I did was say, let's make up a flow field that actually puts the layers back the way I think they started. So I untilted the layers. So this is actual data that make us throw up. Yeah, can you just loop that? So I took data and I just made this, it's a simple kinematic stream function. It has nothing to do with dynamics. And I said, let's see if we can get back what the original patchiness looked like before it got tilted. So I'm trying to, can we just stop it right near the end there? Maybe drag the cursor over to here. Is that possible? Oh, now we're looping? Whoop. Stop it, Okay, I think you get the idea though. So these tilted layers, are becoming steeper. And if you were just to look at, say, this part of it, is there any way of just stopping it right at the end? Yay. You can see that all the layers kind of got vertical at about the same time. And so now if you just happen to sample here, you'd say, oh, I've got a blob of something, and I've got a blob of something else, another blob here. So the question then is, where did the blobs come from so that they could get tilted? Oh, wrong thing. Yeah, back to the PowerPoint. Okay. Next one. Okay, so these were prepared by Alain. This is our study region, and the box shows where we're zooming into. These are VISO sea surface heights, and so they show the mesoscale eddying field. And over here we have what are called finite size Lyapunov exponents. Basically, you calculate these things by taking particles, bunches of particles, and tracking them to see do they come together or do they stretch apart. And lines of high FSLE, which are contoured here, are lines that particles are flowing along and they don't cross. So things converge to these lines and then get stretched along them. So in a sense, these show transport paths of particles, and these particular lines are going in this direction. So now we zoom in, and here you can see the 4 transects of the MVP, and I've been showing you the bottom one here. And then these are the geostrophic currents calculated from a VISO, and you can see that the front that we sampled was embedded in this sort of cyclonic and anticyclonic eddy field. So we have these 2 eddies kind of screwing things together and dragging stuff from up here and squeezing it down into the frontal system between the eddies. And if you look, there's 3 lines of maximum FSLE that come right into that front through those transects. 2 of those lines, this one and this one, come from offshore, and then one of them comes from inshore here and shoots down. And these lines correspond exactly to those layers, which makes me think that each of those layers was associated with one of those lines and came from a very different place geographically. So the one on the inshore side probably originated near shore out here. The offshore ones were probably several hundred kilometers away, blobs of phytoplankton and high salinity that got brought around here, and then these patches that were hundreds of kilometers apart got squeezed together and stretched apart so that they're only tens of kilometers apart within the front. And so they're totally disparate communities that got squeezed together. And so my sort of schematic for this is this, that we have this flow field, this is just a small section of it, driven by these counter-rotating eddies, and then we have patches of high phytoplankton, one from inshore, one from offshore. So this is a plan view looking down on it, and this is depth and cross-front section. And so if we take a section through these blobs, they have some vertical extent, and they might cross several isopycnals vertically. It's just a patch that's embedded somewhere through the mixed layer. And then over time, these patches get drawn into the front, and they begin to get squeezed laterally and stretched along the front. And you can see that here. And because the isopycnals are being brought together, they're getting steepened up. And it's a process called frontogenesis, where the isopycnals get squeezed up. And you can see they get fairly vertical here. And that the front then wants to relax, to release some of that potential energy. And that's what drives the ageostrophic secondary circulation, which is this thing that I've been talking about. And so over time, these things get more and more stretched, and then the ageostrophic secondary circulation tilts them in space. And so you get these layers that look like they've been subducted, when in fact they've just really been stirred. If there was any subduction, it probably happened along the front here. But for the most part, I think it's just a simple stretching, tilting, and juxtaposition of patches that were initially a long way apart. And so I'm looking for a less kinematic, more complex and physically realistic model that we could apply to a system like that to really see whether this hypothesis works. Can we make patches that look like this realistically, or do we require this small-scale vertical velocities to actually subduct things at the front, which is sort of our intuitive guess, but which I don't think is right. So, that's it.
Good, thank you.
Questions, suggestions, help me.
I do have a question. What is the resolution of that transect? Because I didn't catch it. Is it 1 kilometer?
It's about 2.5 kilometers.
2.5 kilometers. Okay.
Between drops.
Yeah, higher resolution would be even better. There might be more layers there that we couldn't quite detect. Yeah, Ralph.
So when you're thinking of a modeling experiment, you would want to essentially put some kind of passive tracer in a field of, of a model that is high resolution, has a frontal genesis. Would that be the idea?
That's the place I would start. Just make kind of a toy model that had some eddies and realistic a realistic eddy field, and then put some horizontal gradients of some tracer in there and see if we can create these sorts of patches. Can we ever create this structure that we see in both density and depth, or do we need some sort of subduction or sinking or something to create these structures? Ralph?
So would your first experiment be a toy model or maybe a realistic simulation, for example?
Whatever's easiest and whoever has the model.
Because the realistic simulation, we probably, between Chris, Art, and I, we can find some nice fields at 200 meters that have, this type of stretching and frontal genesis.
Great. Just need somebody to do it. It would be really fun to do that if somebody had a student or a postdoc. I'm bereft of both right now, but happy to work with someone.
Okay, Rolf?
So this is an example of biological enrichment at a front that was advected to the front as opposed to arose due to frontal processes.
That would be my null hypothesis, yes. And clearly you do get enrichment at fronts and totally unique communities. And my guess is that if you looked at these communities that on these same scales, I would bet that that one, which is the one that came from inshore, and this one that came from north and slightly offshore, are quite different faunistically. That would be my prediction.
Well, that's
Let's
testable.
look at the
Mike?
data if indeed we sampled those with our CTV.
If you have the right resolution, I would love to know. And even in Stuckel's paper, I think there were some points in one of the layers and some points in others that got conflated to be the same layer, and I think that might change your interpretation a bit. Mike had a question. Yeah.
I guess the next question is whether they have any implications for export. I mean, do they remain buoyant, or do they follow the isopycnals down?
So that's where biology becomes interesting, because the phytoplankton, the particles, can move relative to the water. So you could actually, if the tilting then somehow maybe shades the lower part of the euphotic zone, and the cells sort of start to senesce and aggregate, they could sink out. And so it may be that one of these layers is much more prone to carbon export than others. But
Which isopycnals are also going down as well.
well, so that's part of the question, is how much is this sliding along isopycnals and what's driving it and how much would it continue? And I don't think anybody really knows the answer to that, to tell you the truth. Kathy?
So you're offshore, high chlorophyll. No, I mean the patch that you said is a high chlorophyll patch that's coming not from near shore but from offshore.
That's this one.
How do we generate a high chlorophyll patch offshore, or is that just an older originally nearshore patch? I mean, how do you generate? Is it like generated by frontal stuff offshore?
So that's a great question. I can't answer it. It might be exactly what Ralph was just asking about. That might be the patch that it's actually responding to the frontal processes, and perhaps there's some along isopycnal nutrient injection that's occurring here in the frontogenesis region. I don't know. It's obviously a very complex phenomenon. There's lots of things going on, and we haven't explored this in models, so I just can't answer your question. I think those are great questions.
From near shore, but just from different places and different ages or something?
Well, this suggests that the two offshore ones actually came from offshore, and the nearshore one, the onshore one, came from closer to shore.
What does the satellite chlorophyll say in the days preceding?
I don't know. I'm not sure we could even see it because of clouds and things. I'm not sure. I can't answer that. But I think looking at them faunistically, I think would be really interesting and revealing, especially if there are any samples from upstream. I think there's lots of interesting biological implications, and these are excellent questions, and I just can't answer them. It would be fun to have a good model that would...
Thank you for sowing the seed.
Sure.
Great, and so I think we'll move on to
Good
Jessica
idea.
Liu and
How
Matt
far
Long.
offshore was that, like 300 kilometers?
No.
About 150. By the way, I'm thrilled to have other people thinking about my MVP data, but I appreciate that they didn't forget whence the data came.
Thank you, Mark, who was a co-author hidden from this time. I have to rush out. I'm sorry for all such things.
I think if you could wait for just the introduction to Jessica's presentation, I don't know if you have a couple of minutes for that.
My class starts in 5 minutes. It's all the way down the hill.
Okay, so Jessica and Matt, you're up.
Hi. Hello.
Hi.
Can you guys hear me?
Yes.
Yeah. Thanks, Mark, for letting me present here. I was hoping to catch Peter before he left, so I think I have 5 minutes. But this is ongoing work. This represents maybe about 6 months worth of work, so very much a work in progress. I'd love any feedback at the end. And this project is size structure plankton modeling in the community Earth system model. And I'm a postdoc at NCARD, National Center of Atmospheric Research. So within the context of biological oceanography, two of the major challenges are predicting climate change impacts on marine food webs, as well as on the biological pump. And Debbie Steinberg and Mike Landry had this nice schematic in their annual reviews paper, which shows a lot of complex processes happening. What's really important here is that carbon export models need to be linked in with food webs. So for the global domain, food web models are often difficult to generalize, and another type of models are often used. Bioenvelope models cannot take into account feedback loops between the biota and the physical, like the climate system. Within this context, we wanted to account for the size of organisms and actually size of sinking particles, because that would affect the transfer efficiency to depth. So the first part of this effort is using size as a master trait for modeling marine organisms. Size-based models have been used in a lot of different contexts, from individual-based fisheries models to plankton models, and Peter has done a large number of work studies on this. But what we're hoping to do is just focus on the lower trophic levels, from the phytoplankton up to the mesozooplankton, and represent size-based allometric relationships in a global biogeochemical model. So what we're using is the Marine Biogeochemistry Library. And the Marine Biogeochemistry Library is a relatively new formulation of existing model that's used within the Community Earth System model, the BEC. And MARBLE, as it's abbreviated, is designed to be modular in nature. So it's designed to be able to plug in and out of a number of different ocean GCMs. Right now it's formulated to be within POP, but we can, there's ongoing efforts to put it into ROMs, or the future in MOM model as well. Within MARBLE, the default model includes 3 phytoplankton and 1 zooplankton, with different tracers for chlorophyll, iron, silica, calcium carbonate, phosphorus. And the 1 zooplankton is this adaptive zooplankton that feeds on the different types of phytoplankton, but really just represents maybe just kind of a glorified mortality term for the phytoplankton. So what we wanted to do is extend this idea out a little bit further of perhaps providing flexibility in the construction of the ecosystem as well. So we've done a bit of work here at NCAR to enable the flexible, flexible ecosystem construct. So we can put in, we can have a default setup, or we can have any number of phytoplankton. If you wanted, or zooplankton. If you wanted to run with, for example, 100 phytoplankton and 2 zooplankton, you could do that. But in my current example use case, this is kind of a proof of concept, I've developed a model with 9 phytoplankton and 6 zooplankton. So I have 3 diatoms, 4 mixed phytoplankton, a picoplankton, and 6 zooplankton, as well as the diazotroph group. This model uses the concept of allometric scalings to generate all of these variables. So for phytoplankton, here's what you see on the screen here is just a list of the various different types of parameters that go into the model. And with relationship to size, I've just indicated in a simple arrow, but some of these slopes vary quite significantly from each other. So in general, maximum photosynthesis rate decreases with size. the initial slope of the growth irradiance curve, which is a metric of how fast a phytoplankton can respond under low light conditions, decreases with size. Overall chlorophyll A to nitrogen ratio decreases, iron to carbon ratio decreases, and then phosphorus to carbon ratio increases. A bunch of half saturation constants increase with size, and this has to do with, as you increase the size of a cell, there's more area upon which different molecules can be transported in and out of the cell. But there are some key exceptions that we had to build into this model. So for example, picoplankton, Prochlorococcus and Synechococcus are known to have really low photosynthesis rates for their size. Diatoms have high chlorophyll A to nitrogen ratios. And then there's some evidence within kind of this trait-based literature that suggests that diatoms may have a shallower slope for half saturation constants. And this is really because of their internal vacuole. Essentially, the thought is that they are mimicking a smaller cell in a larger cell's body. And for the zooplankton, there are 5 key parameters. Grazing rate also decreases with size. The half saturation constants I have in my model as decreasing with size. I pulled this from actually Taniguchi et al. 2014, a data compilation. Respiration rate, this is the metabolism increasing with size, mortality decreasing, and fraction of losses to try this increasing with size. And then finally, what I have as well is I represented the predator-prey feeding relationships with a Gaussian feeding kernel. So zooplankton would have a preferred size prey, but also be able to feed on a number of other size prey according to a Gaussian relationship. So here's just some initial results. This is pretty hot off the press, but in a 62-year hindcast and a 1 degree ocean model resolution, this figure on top shows the total surface chlorophyll, and the contour lines mark the values of the chlorophyll. So you can see that we are able to represent the low productivity in the gyres, relatively high productivity along the high latitudes and the coasts, including the CCE. And we have a pretty good value for net primary production, about 61 petagrams of carbon per year. So I was pretty happy about this, just to be able to get the biogeography right. In terms of export flux, my current model is actually on the low side, so I'm getting about 4.7 petagrams of carbon per year. Our current estimates of export flux ranges by a factor of 3 from Henson et al. Estimating 4 petagrams of carbon to other estimates at 13 petagrams of carbon. So we're within this range, but definitely a little low. And I have some. This is ongoing work, so I'm trying to figure out how to fix this situation. And we're also able to prognostically generate global stoichiometry. So this shows a map of global surface phosphorus to carbon. And this value also is a little bit low. It should be between 10 and 11. But I'm still able to represent the general biogeography patterns. And then what's most interesting for me is a prognostic global size spectra slope. So what you see on the right is your normalized biomass size spectrum with all phytoplankton and zooplankton together. There's some bumps in the curve, but the the mean values for the surface are definitely within the reasonable range for what you would expect in the global oceans. So in comparison to, going back here. In comparison to some data from the Atlantic Meridional Transect from San Martin et al. 2006, these are 3 passes along the same transect, and there's obviously a fair amount of noise in this data, but generally they have a lower. Sorry, what I've done here is I've reversed my sign convention to match theirs. So they have slightly higher values in the lower latitudes and a lot of variability in the higher latitudes, and that's roughly what we see here. And then this slide that keeps popping up is just showing the seasonal cycle in terms of an anomaly plot. And what's fairly interesting just from first glance is you can see actually some shifts, for example, in the high latitudes, seasonal succession. So going from April through to August, you can see the progression of blooming different size plankton just by the sign of the size spectra slope. So I've plotted here, this is just for the Arctic region, just as an example, the size spectra slope from surface to 150 meters from May through August, and you can see that in May the size spectra is pretty close to 1 all the way down to 100 meters, but then there's you can see the signal of different phytoplankton groups blooming in succession that pull the slope of the size spectra to more and more steeper values or higher values as the summer goes on. So that's just an example of the type of data that I'm able to get from this model, and I'm really interested in looking at how, for example, the size spectra may change with day versus night, with different seasons, and with different physical features, and not only perhaps looking at the actual magnitude of size spectra changes, but maybe the error, the RMSE of the regression model fit, because that may indicate a relationship of plankton community dynamics to their environment. So in summary, the size structured plankton model can be a parsimonious method of adding ecosystem complexity. So in here, the allometric relationships were key. And this model enables future integration with size-resolved detritus groups. And there may be potential for development into a continuous size-based model. This might be of interest to some of the people in this room. The plankton size axis is definitely separate from biogeochemical function. So within a PGC model, you would need to have nitrogen fixers and calcifiers and silicifiers. But not only that, key groups such as diatoms may have different nutrient acquisition needs that would need to be taken into account. And then other exceptions would apply for example, low picoplankton growth rates are essential here for my model. The zooplankton feeding kernels can be used to generalize grazing relationships. So right now what I have is just one single function, a Gaussian function, but you can imagine adding in other zooplankton with that are perhaps specific versus generalist predators, perhaps a SALP in there to represent filter feeders, there's a fair amount of potential here. The plankton size spectral dynamics are definitely an emergent feature of the system, and I mentioned this before, but I think that there are a lot of interesting patterns that we can pull out of this kind of model. I still have a lot of outstanding issues before we get a model that it can be put into production. So one of the key things is within our current model structure, there's no zooplankton growth, and that may become an issue. I still have low POC export flux that I need to fix. And then one question is how to deal with exceptions to allometric rules. How do you deal with, for example, a jellyfish that doesn't follow by standard allometric rules? And I'm not entirely sure how to deal with that, but that would be a topic I'd be interested to discuss with anyone. So anyway, that's it for me.
Okay, well, thank you, Jessica. I'm sure she'd be very happy to receive comments and feedback.
Yeah, yeah, any questions?
I have a question.
So this is Madi Karu. You have to speak up.
Yeah, there was a very interesting map of this global map of the slope of the size spectra, and there was a big anomaly in the Southern Ocean, just west of South America. This is the one, just west of South America. And how do you explain that?
Oh, this big anomaly here. So this, I'm not entirely sure. Within the context of the Southern Ocean, there's a lot of structure in this area. This could be, in general, when there is a very large slope, so steeper slopes, that indicates a dominance of picoplankton and nanoplankton. So it could be that in terms of like an annual mean, this area, the model is producing high picoplankton and nanoplankton in this area. Yeah, but in general, the Southern Ocean had a lot of variability and a lot of structure. So one of the things that I'm going to do next is not only look at this mean value of this mean slope, but also how it varies, and then the RMSE of the model fit, essentially, to the data.
So Jessica, I actually had a question about another part of this figure. So there's such a striking difference between the southern and northern Atlantic in the size spectral slopes. Why would that be? And that doesn't correspond to what showed up in the AMT. I realize AMT did not go to the poles, but
right.
What's going on there?
We're talking about basically like this area versus this area.
I'm looking at the lower left panel, the slope, the blue line.
The lower left panel, right. Oh, yeah. This is very curious. The AMT didn't sample that far south. So my interpretation of what this means is that, again, this shows the influence of some of these high values, so potentially dominance of pico nanoplankton, and versus I would say when you're looking at this very high latitudes, this is kind of getting into the Arctic area. So I'm not. And also the way that I've constructed this region mask, it's actually averaging over the entirety of this high latitude region. I don't have a good answer, Mark. I'm sorry to your question. This value of slope definitely indicates that there potentially would be larger organisms, so potentially larger zooplankton, and larger diatoms, larger protists. Oh, sorry. I was just going to say that I was actually within the context of putting together these slides for the CCE Forum, I was looking for some data for the CCE region. So I was wondering if anybody knows of any size size spectrum data for CCE, feel free to send it my way.
Actually, Tristan Biard and I are working on exactly that, so we should be able to help out.
Oh, okay, that would be great.
Just a comment is, in the mesozooplankton world, we tend to see very large organisms in both the southern and the northern hemisphere polar regions.
Right, yeah. So then it is a little, it is a little bit strange that we wouldn't have, yeah, that we wouldn't have the signal of the large mesozooplankton in the Southern Ocean. Sure. This is, I mean, I would definitely still have a fit to dig in, and this is not a finalized model by any stretch of the imagination.
Okay, well, certainly it's an interesting approach, and I like the fact that you're taking an allometric approach, but with exceptions. I think that's a sensible way forward. You said that you assigned the maximum grazing rate to decrease with body size of the zooplankton, and so you must mean the mass-specific rate.
Right, right. This would be, yeah, right.
Okay, thanks you. That clarifies. So any other? Yes, Jessica.
Hi, Jessica. Nice talk. We met in Ocean Sciences at one point. I was just wondering what processes you include for your export.
Oh, so so right now in the current model, the export is just is not explicit. It's really just an instantaneous redistribution of mass that happens according to a remineralization length scale. So this would be potentially the next step of this project once we get the food web working and reasonable and defensible, is to add in a explicit export and tie in the size of the zooplankton, for example, to the size of a particle being exported, and thus the speed of export. So right now what we have is just, it's not an explicit detritus group or an explicit export.
I think when you do your carbon export's going to go way up.
Mm-hmm. Potentially, yeah.
Okay, other comments? Okay, and Jessica will be at the Ocean Sciences meeting if people want to follow up and talk with her further. Okay, thank you so much. So then Chris Edwards, coming in from UCSC on biophysical data assimilation studies at UCSC.
Hi, everyone. Can you see my screen?
We can see your screen.
All right. It's been really interesting hearing all of the talks that you've put together, and there are a lot of different scales that are represented. I think the work that I'll be talking about is kind of like Bruce's, somewhere in between the different scales that Manu was talking about. And what I'll be talking about is a collection of modeling activities that are going on up here at UCSC, and there are a lot of collaborators. In particular, I'd just like to highlight Andy Moore and Paul Matern. Jerome Fichter is also involved in a lot of the biogeochemical modeling activities, but not represented in what I'll be talking about today. Anyway, hopefully this gives a good idea of some of the things that are going on up here. I'm going to mostly talk about biogeochemical data assimilation efforts that are going on, but I'll start with this description of some physical reanalyses that are being carried out mostly by Andy Moore, some by me. Our standard model domain extends, as is shown in this figure, from mid-Baja California up to Vancouver Island nearly, and offshore quite far. We have a number of different models that we look at, including the ones that Manu talked about earlier at kind of high resolution nonlinear forward models. But all of our data assimilative efforts are really being done at 1/10 of a degree resolution. That's pretty coarse, and we don't include things like tides or rivers, but we've done a lot of work on this. We use ROMs. We put together a number of physical reanalyses, 31-year reanalysis going back to 1980, a 12-year reanalysis that started in 1999 when a high-resolution atmospheric model came to exist. And in support of IUS and SENCUS, we run a near real-time system from 2011 to the present, running with that COAMPS atmospheric forcing. The physical models have been used in a variety of studies for a lot of different purposes. For example, here's a study that Mike Jaycox, when he was a postdoc here, performed looking at upwelling along the coast of California. What I'm showing on the left is model vertical velocity across 40 meters depth when averaged from a 31-year reanalysis. So this is a state estimate, a lot like what Bruce was talking about before. It incorporates all available data from 1980 to the present. Obviously, we missed a lot, but we included a tremendous amount of in situ and satellite information. You can see the strong upwelling signature along the central California coast, and you can do things like evaluate whether there's good agreement or not with simple ideas about upwelling, such as the Bakken upwelling index shown in the upper right. What Mike found was that the agreement was pretty good. The model W is shown as the red curve here as a function of latitude out to about 200 kilometers from the coast. And the black diamonds are the coastal upwelling index that's produced by NOAA in basically using Ekman transport ideas and an atmospheric model significantly averaged. You see that at latitudes south of about 39, there is a growing disagreement between the state estimate produced vertical velocity and that predicted by the upwelling wind stress. And Mike was able to show the reason for this discrepancy had to do with onshore geostrophic flow that's part of the larger scale gyre circulation that contributes at these latitudes towards offsetting the total upwelling from what one might expect from the wind stress alone. Anyway, I guess my point in showing this particular study is that the reanalyses are useful for producing data-constrained estimates by which you can test ideas that we think help us understand the ocean processes. So the rest of my presentation is going to be about biogeochemical things, and the work that we've done has primarily used two BGC models. One is a simple 4-component NPZD model, and the second is a more complex 11-component NEMRO model. We started simple and then moved to a somewhat more complex one, simply to understand, well, in part to understand the sensitivity of things to greater phytoplankton diversity and zooplankton diversity, as was being talked about in the last talk, but also because we think that the more complex models are really better representative for the California current, as I'm sure everyone in the room would agree. We do some non-data assimilative parameter estimation efforts, and what I'm showing in the lower panels here is a comparison of unoptimized ecosystem model versus optimized ecosystem model output relative to data. So in the lower panel on the left is some data. It's a one-year run, it's a one-year average showing chlorophyll as estimated from CWIFs. And then there's sort of a first guess using the NPZD ecosystem model, and you can see quite a bit of discrepancy between the reference case and the data. And through an objective cost function that's based on both in situ chlorophyll and climatological nutrients that I'm not going to go into here, we find we are able to optimize the parameters, many, many unknown parameters that are poorly constrained, can be constrained to some extent by the available data in a realistic simulation. So I'm showing two efforts at optimization. The first one is in the third panel on the bottom, and that comes from a Monte Carlo simulation where modifications of the parameters get a little bit closer to the CWIFS data. And then on the lower right is a further improvement associated with a genetic algorithm, actually a sort of a constrained search procedure. And I'm going to just show this figure, which highlights how these biogeochemical models, even simple ones like NPZD, are challenged by the many unknown parameters. Even in simple models, there are lots of things we don't really understand. And you can look at these optimization procedures to try to look at biases or consistently poorly represented parameters. And I guess what I'm showing actually is that both NEMRO and NPZD show a variety of parameter combinations that yield effectively the same cost function output, which simply means that we're dealing with nonlinear differential equations that do not have simple parameter space minima. And we should sort of expect that as we do all sorts of experiments with these types of models. All right. I'd like to go on now to state estimation, and this is a lot like what Bruce was talking about. I think the main difference is that in addition to doing it with physics, we're also now doing it with these NPZD models. And this is an example of a twin experiment. So this is completely in model space where we are improving biogeochemical state estimates through assimilation of biogeochemical data, but we're also assimilating physical data at the same time. And what I'm showing in these plots is simply that the lowest RMS error comes from combined physical and biogeochemical data assimilation. The physical properties are constrained mostly by the physical data. The biological properties are mostly constrained by biogeochemical data, but the best output comes from doing both. We find this is true in model twin experiments, and we haven't really demonstrated it to be true in realistic circumstances, but in principle, if we have a good biogeochemical model, it should be true. All right, I'm going to go on now to a figure that illustrates what we're doing. So on the left panel are 3 time mean averages. We have data, it's a 1-year average surface chlorophyll from CWIFS. And on the right is a Hofmüller diagram with latitude on the y-axis and time on the x-axis. And it represents an average of the chlorophyll between the coast and this white line here, which is 200 kilometers from the coast. And one thing that clearly comes out is a lot of white space in the Hofmüller diagram, which is indicative of no data being available at that latitude at that time. And there's a lot of data loss due to cloud cover, which is a problem that we can think of the state estimate as being one way of filling. Anyway, if we go to the second row, we see the model with and without, with the model with no assimilation. And this is a NEMRO model, and what you see is standing stock that increases at about the same time as it appears in the data. However, there are clear discrepancies between the free run and the data, there are lows that are too low at some latitudes, there are highs that are too high, and the timing of the exact bloom details are off. So with a simulation, we can constrain that estimate of standing stock to be consistent with the data, and at least within short assimilation cycles, we use 4 days to be consistent with model dynamics. All right, the last thing that I'd like to talk about is, I think I'll skip this. We can talk about forecast skill. The last thing that I'd like to talk about is some work that's not published, but is sort of lurking in the background of what we're doing. We recently had this anomalous period of the blob moving into El Nino in the California current, and we performed a biogeochemical analysis of processes that were active during the blob. So this is a 4-year simulation, and what I'm showing in the upper left panel are domain averages of surface temperature, and I'm simply showing that the discrepancy between the blue line and the red line show that we had the blob. We can express it in terms of the time series of temperature or in terms of the SST anomaly, which reached 2 degrees, or we could represent it as a standardized anomaly, and we get up to 3 standard deviations from the mean. And it doesn't matter what averaging window we pick. For example, this large region over most of the CCS, or maybe a smaller region that's closer to the Central California coast, we had the blob in our reanalysis, and we can define a period of time to focus on, maybe 2014, 2015. We can look at the dynamics. If we look at standing stock, we have 4 years of standing stock, and you can see that some years, like 2013 and 2016, had very high nearshore standing stock in our reanalysis, whereas the years of the blob were anomalously low in their annual average. And we can focus on processes. Now I'm just jumping to the kind of the end results here. If we look at summertime averages, we can break down the phytoplankton budgets into physical processes like advection and diffusion, and growth terms in the dynamics and losses. And if we do that, we get a simple balance between really biological growth and losses. The physical processes are very important, very near shore, but they're still small compared to the biological processes. We can do a much more detailed deconstruction of the different processes and then do a comparison between different years. So I'm doing the comparison here, and what we find is that the years of the blob were unusual in that they had particularly low new production. And so this suggests a bottom-up control for the anomalous standing stock during those years. And we can diagnose the processes that are underlying that change in new production. So this is a busy plot, and I think I'll just jump to the point, which is that in this 4th panel, you can see a time series of new production integrated out to about 50 kilometers from shore. And what we found was that springtime production during the blob years was not actually all that low. What was really low was summertime production. So the summertime months were unusually low, and that was not the case in pre-blob years, and it was not the case in the El Nino year as well. So we can then try to diagnose what was causing this, and we can look at whether it was a preconditioning of the nitrate along a certain isopycnal. We didn't find that to be a large contributor to the total variance. Instead, we found that vertical velocity, say at 30 meters, was the strongest contributor, and that is partially explained by the wind stress, about 67%. So I think I'm going to stop there. I think what I tried to do was just give a description of the types of different studies that we're doing in the California current, mostly with data assimilation, and they involve kind of technical details of data assimilation, but also trying to look at basic processes of scientific interest. Okay, thank you very much for listening.
Thank you, Chris. Lots of interesting work going on. Comments, questions? Chris, I had a couple questions. So one, concerning your diatom budget, I like the way that you decomposed the physical and the biotic processes, yeah, on the right side of the equation. And then this is a sensitivity analysis as a function of distance offshore, but of these four basic processes, it's more than four, of the basic processes, which do you think are the least constrained? In other words, in terms of providing advice from your modeling efforts to the measurement people, what do you think needs to be resolved much, much better?
Oh, that's a great question. Thank you. So I'm going to go back to this figure. So in some sense, this is maybe not very satisfying, because we are in fact assimilating the top panel information. And if I understand your question best, you're saying, what's the information that we need in order to do this better? I'd like to say everything, which is not very helpful. We work with 4 or 11 component models, and we have data really for 1 or 2 boxes. I think subsurface nutrients on gliders are going to be a game changer for this type of work. So that is a high priority in my view. I think other pieces of information would be really excellent. Over the years, Mark, you and I have talked a little bit about zooplankton data. It would be great to have a form of the Cal-Coffee zooplankton record in the same currency as the biogeochemical model. So I know often the Cal-Coffee record is in terms of counts, and we just need a useful conversion of that to biomass. I think that will be another incredibly important element. The other parts of the biogeochemical models could be constrained as well, but those two, subsurface nutrients and zooplankton, something about the grazers would be fabulous.
Okay, that's helpful to know. Also, a comment about your inferred changes in new production during the blob years. You're probably aware that Todd Martz has utilized the CC2 mooring data to infer changes in the F ratio, and making a steady state assumption, which is actually not particularly good in the upwelling area. But in any case, there should be continuous data that would enable you to infer the F ratio to see how well it matches up with that 4th panel new production curve.
That's really great. So you can see I separated out recycled production from new production, and there will be changes to the F ratio, but it honestly didn't occur to me to plot that. So can you give me the name again that you mentioned? I am not familiar with that work.
Yeah, it's Todd Martz, M-A-R-T-Z. He's a marine chemist here. He's developed pH sensors and pH oxygen sensors, and collaborates with Uwe and me on the CC2 moorings. I'll send you the reference to the particular F ratio paper I'm referring to.
That's great. I really appreciate it. And lastly, I'd like to just advertise that we're looking for a postdoc up here who might be interested in some science applications of the biogeochemical modeling. So if any of you know of people looking for postdocs, we'd like to hear from them.
Good, we'll spread the word. Okay, Mike Leandry.
Yes, I'd like to follow up a little bit on Mark's question about the diatoms. And the question is, does it make a difference in your model what kind of diatoms they are? Because I think one of the things we noticed during these warm water events is that we hit some very different types of populations or species, whatever. And so do you have like a fixed parameterization for diatoms, or that vary?
We do. Thanks for that question. We do, in fact, have a single diatom in this model, and it is parameterized. Well, we can find that we could have many different types of diatoms with reasonably similar bulk statistical performance. If you asked me what particular diatom was used in this study, I honestly can't. I would have to go look at that. So it is just a single one, though. It's not representing an ensemble of diatoms.
All right. Yes, and Jessica Carrier.
I was just wondering what biological parameters or other parameters that you're assimilating that improves the physical parameters, like the velocity.
Okay, thanks for that question. So this figure shows only a model twin experiment, and let me just go through this more slowly. So in the upper panel, I'm showing the RMSE for a particular experiment we did, looking at 30 one-month runs, and a free run gives this RMSE in velocity, assimilating physical data in the same way that we assimilate sea surface temperature, sea surface height, and some in situ temperature and salinity. So the data was mimicked to be realistic based on positions and types of real data, but it does come from a model. Anyway, with a physical assimilation, we get an improvement over a free run. And when we do biological assimilation, so here we are just assimilating chlorophyll. And if we don't assimilate physical data, then you can see relative to the free run, we actually get an improvement. And that's true for both velocity and for sea surface height. However, you can see that the biggest improvement come from assimilating physical data. In the reverse, when we look at the biological fields down on the bottom, we get a little bit of improvement by assimilating ocean currents and sea surface height and sea surface temperature, but we get a significantly greater improvement by assimilating biogeochemical data. So it's not hard to imagine how these things work inside the model. If you improve the physics, you improve the positions of the eddies, and you should get better representation of the responding biogeochemical field. In the assimilation procedure, the information that comes from surface chlorophyll tells us something about where the eddies are also. And so there is an adjustment that takes place through assimilation where the physical properties are improved as well. Does that help answer your question?
Okay, very good. And now on to Natalie and Art on another synthetic presentation. Natalie Kiros and Art Green. And is there anyone else who I didn't have on the list who wanted to make a presentation in the room, or anyone else who's not in the room who had something they wanted to contribute after Natalie and Art's? Okay.
Thank you, Mark. Hi, everyone. I'm Natalie. I'm doing my PhD with Art Miller. And I'm going to present the results of a work we've been doing in collaboration with Matthew Long and Jessica Luo on building up a composite physical-biological reaction of California current system to El Niño. So this is the experimental setup and the model that I'm using. It's based on CSM version 1, and the oceanic component is a POP-CIs, version 2 and 4 for the CIs component. And this is based on the core protocol, so the forcing is going to be based in atmospheric reanalysis. Also, it has embedded a biogeochemical component by the Beck biogeochemical and elemental cycling. I cannot see a pointer, is it?
Yeah, pointer doesn't work on a TV screen.
But you can use the poll over there if you wish.
Yeah. Okay, so it has embedded the Beck model for the biogeochemistry, and the results that I'm going to present are based in the California current ecosystem. So what I did was take all the El Niño years and I divided them into the precursor year, which is on the top, and the peak of the El Niño year, which is at the bottom. So these are SST anomalies for the simulation. Then we can see the composite of the precursor year, and then below we can see how the anomalies peak during February, March, a little bit in April of the El Niño year. This is the same, but taking the La Niña years. So we can see also this is the fall of the precursor year, and then we can see how the anomalies peak in January, February, March. So yeah, this basically is showing the cold anomalies related to El Niño in California current system as a response to El Niño, or La Niña in this case. Can I change it? Yeah. Okay. And we did the same for the thermocline depth. So for the thermocline depth, I took the 26 potential density, its depth. So it's the same, we're looking at El Niño, the precursor year, the fall of the precursor year, and then the winter of the actual El Niño year, where the thermocline depth anomalies peak during February, March, and April. So here red means positive anomaly, so it means a deeper thermocline with respect of the climatology. I forgot to mention, I'm sorry, that the climatology is from 1948 to 2015. And we have the same for La Niña. So we can actually see how, well, La Niña is pretty flat actually, but we can actually see how the positive anomalies of the thermocline depth start developing from December, and then they peak in February, March. So again, the negative anomalies here means a shallower thermocline. And we can see that these anomalies persist through April, and I'm not showing it here, but they persist through the summer of the La Niña year. And this, so I took different locations. I don't know if you can see the red points, but so upper panel is the northern points, and then the southern one, and this one corresponds to the second point. So this is the composite of the thermocline depth, but taking only the second year, so it would be this year. This year for El Niño, La Niña, which are La Niña in blue, El Niño in red, and neutral years correspond to the black line. So we can actually see that during La Niña years, the thermocline is shallower, and for El Niño years, the thermocline is deeper. What I think is interesting here is that this figure actually shows where these anomalies peak. So we can see that for the northern points, which are above, these anomalies peak more during the fall, which is September, October, November, and then for more central California, we have that these anomalies peak more during the summer, which corresponds to June, July, August for both of the points. And this is showing that the model seems to be a little bit delayed with respect to the observations, which, I mean, we know that off here, these anomalies peak during the spring, which around March, April. And then off the Oregon coast around here, they peak more towards the summer. So this is a little bit delayed. And then I'm going to move on to show some chlorophyll composites. So this is just the biological features of the model. It includes 3 phytoplankton groups: diatoms, small phytoplankton, and diazotrophs. And for the composites, what I did was taking the vertical integral down to 100 meters, and then down to 25 meters to compare with observations from satellite. And then I also did some calculations of the carbon chlorophyll ratios over CCS. All this is for the model only. Okay, so we start with the chlorophyll anomalies composite for El Niño year. And this is only this is going to be all of them, the precursor year of El Niño. So we can see how, I mean, this El Niño hasn't kicked in yet. So this would be the normal behavior of the CCS during summer. And then we see how the anomalies start going towards the negative side when El Niño kicks in. And then we see that during the spring, and summer, we have negative anomalies for the chlorophyll, and they persist through all the year, through the summer, and then the rest of the year. And then we have the same for La Niña, and yeah, I mean, again, this is when La Niña hasn't kicked in, and then we start seeing some positive blooms of the California coast, and then we see how the positive anomalies peak again late spring and persist throughout all the summer of the La Niña year. This is a comparison from the model, the seasonal chlorophyll. This is not anomalies, it's just seasonal means. From 1982 to 2010, which is the range available for the satellite. So above we have the model, integrated only until 25 meters, and then above we have sea waves. And well, we can see that the chlorophyll seems to persist pretty high on sea waves, and then even in fall. And then in the model we see that the chlorophyll the values are very low. And then this is the carbon chlorophyll ratio for, again, seasonal. So we have winter, spring, summer, and fall. And this is for all the years, not taking neutral or Niña, it's just all the years for the whole period of the model, 1948, 2015. And these are all the points all over the domain of the California current system. So we can see that high ratios represent dominance of non-diatom. So this R is a carbon ratio of non-diatom versus diatom plankton. So we can see here that higher ratios mean non-diatom groups are dominant. But we can also see that when smaller ratios we have, it means that we have more diatoms, we have more chlorophyll. And this is true for all the seasons. What else here? Well, I mean, also here that we have so the scale is different. So this means that we have more diatoms during spring and winter than we have during July and during the summer and the fall. And some of the conclusions are, as I mentioned before, that the model shows a delay from 3 to 5 months in the thermocline depth anomalies. So of the Central California coast, they're observed to peak more in summer, when we observe that they typically peak during the spring. And then off the Oregon coast, they peak into the fall, and typically they are supposed to peak in summer. And the SST anomalies are observed to have a delay only from 1 to 2 months, peaking instead of December, January, February. They seem to peak in February, March, April, but I think there are some El Niños where even in the tropical Pacific, the anomalies peak during this season. So I think this is just an artifact of taking all the El Niño years and averaging them together. But yeah, I will see. And then, yeah, we have seen that the temporal variability of the chlorophyll anomalies is consistent with the variability of the thermocline depth and also the SST. So we have that when the thermocline is deeper, then we have less chlorophyll. When the thermocline is shallower, we have more chlorophyll. At least, even when the time seems to be a little bit delayed, the model is consistent within. There's consistency within the model. That's it. Thank you.
Thank you. Okay, questions?
So this figure was motivated by Peter Franks, who said to plot it for the model compared to what's observed, at least at some of the stations that he's looked at. And his plots tend to show basically for low chlorophyll, basically they just go straight up and down, right? And then for high chlorophyll, they just go straight across. So it looks almost like L instead of this kind of gradual slope that the model was producing. On the other hand, these are distributed over the entire California current, and his is more localized right in the upwelling region. So when you get the big blooms, they tend to be dominated by the diatoms, and then if you get the small low chlorophyll, they tend to be dominated by little guys. So it's much more severe than the model was produced. Obviously, it's limited in the types of critters that the model has, but it was an interesting idea to look at this.
So it was Peter's analysis of your HPLC data, Rolf, or the size fractionated data?
I don't know which analysis.
I forget the citation. Do you remember that?
Lee? I don't remember.
Yes. Chen. Okay, Chen Lee. That was part of the A-front modeling study.
Yeah. It wasn't more. It was based on microscopy, I think.
It was the observation.
So based on your microscopy.
It was the observation.
So what is R in here? Is that the chlorophyll ratio? Carbon chlorophyll
this
ratio?
one?
No, can't be.
Yeah, I mean, because this is carbon non-diatom and diatom carbon ratio, and this is chlorophyll.
N over D, right?
Right, so N over D is non-diatoms and diatoms carbon from the model.
Okay, so those are carbon on one axis and chlorophyll on the other. There's no, the ratio is implied.
The ratio is on the left. No, the R is the ratio, right?
Oh.
Non-carbon to...
You have a ratio
non-diatom to diatom.
Right.
You have a ratio of 2 or 5?
I think it's two different estimates of carbon or ratio. Plotted against chlorophyll, and the slope then is the carbon to chlorophyll ratio.
Yeah.
Yeah, okay.
So have you considered separating Eastern Pacific from Central Pacific El Niños?
Yes, but I haven't done anything yet.
Well, the first thing is, I mean, I guess Matt Long is listening, and probably Jessica Luo is also, unless they've already checked out, but hopefully we can talk at the Ocean Sciences. The first thing is, when you use this particular model, it's only 100-kilometer resolution, right? So we're not resolving a lot of the upwelling processes right at the coast in terms of the physics. But it is using observed forcing, right? So it's a bit unsettling that when you just look at the physics itself and compare it with available observations, there's a fair amount of discrepancy, even for the big El Niños. And if you add in also the smallish El Niños or middling El Niños, it's even more not particularly good physically, right? So then you take that physical model, and now you're driving an ecosystem model, and it's also having issues representing even things like the proper timing of the spring bloom in different parts of the California current. So there's a cascade of disinformation that's making it a little difficult to do what we originally thought to do, which was to do sort of a physical biological composite of El Nino for the California current using this particular model. So we're going to have to take a slightly different tack in sort of explaining what's useful to get from this than just saying simply we're doing that. So to supplement our understanding of the way models function, we've already gotten a separate run from Enrique Richitzer that is, 10-kilometer ROMS regional model, right? And that one has both physics and biogeochemistry in it, although it doesn't have the oxygen. It stops at just nutrients, right? This one actually has things like oxygen.
Yeah, this one, yes.
So we can do a little bit more from the biogeochemical perspective with this one. And then I don't know what's going to happen when we do the analysis of the ROMS one from Rutgers, but we might get, I don't know how similar these things are going to be. But it's kind of an interesting exercise just to look, A, that obviously when you run a model, you don't get a perfect physical state, and B, when you don't have a perfect physical state, the ecological state is even more in question.
Well, I was sort of naively hoping that if there's, let's say that there's a difference in timing of arrival of equatorial Central Pacific Ninos and equatorial Eastern Pacific El Ninos, that if you were to then separate the climatology of CPs differently than EPs, that it might improve things like the timing of the thermocline depth anomaly, et cetera. So that would that not improve matters? I mean, Laura is working to some extent on this, asking the question of whether CPEP matters for the zooplankton.
Right.
And there's some evidence.
So that's doing purely in nature. I think what you're saying is reasonable, but in thinking about it from the perspective of this particular model, since it doesn't reproduce nature locally on the California coast as well as you would like, it's a little dicey then to try to separate those two things out, because it's really just the local response to whatever the specified winds are and heat fluxes, right? So the model does not have any atmospheric modeling issues. It's just the actual forcing. So it's not the one. And then the other thing that we wanted to do was to take the El Ninos that pop out of the fully coupled version of this. So you'd have an atmosphere and 100 or 200 or 1,000 year run, whatever it is. So that would just intrinsically generate El Ninos in the model, right, in the tropics, which would then teleconnect to the mid-latitudes and drive ecological and physical changes in the California current in that case. So I think we started looking at that at the beginning, but then we decided to stick with first doing the, knowing the forcing in the atmosphere and driving the model and seeing what it does first, and then maybe going back and looking at the fully coupled case. So this is an example. The biggest El Nino events are shown here in observations and in the model. And a point off the coast, essentially. Is it an average box or is it a single point?
It's a point here in the pier.
In both the model and the observation?
Yeah, yeah.
Oh, so at the pier?
Yeah, so the blue one is a pier and the dashed black line is a model. And I was thinking that maybe the timing can be fixed if we divide the CCE in regions, because I mean here, at least for the 82, 83, and 97, 98, the model peaks in December, January, February, also here. This is a different case, but so we are seeing in the field that in general it peaks a little bit later, February, March, April maybe, but maybe if we average over a small area or we divide our northern CCE and southern CCE, we might get a different pattern, but I'm not sure.
Does it make sense to use the Scripps peer record for a model with that resolution?
It's correlated to almost an area average. If you did the correlation of SIO peer with SST, you just see a big red thing all along the coast. So it would look very similar.
Well, the correlation is especially good in the winter. It's not quite as good in the summer.
But you can see that the model has a muted response. It tends to be lower amplitude response in temperature. And obviously then the chlorophyll itself was also lower than nature.
We've come up against this issue about spatial partitioning of the CCE domain a number of times, and Rolf has one 3-part partitioning that's been useful for summarizing some of the process groups data. Madi has it's a 12-grid system, and it may be useful to try to
Mine is not a spatial partitioning, but you can like it.
Well, it's a hydrographic partition, but the hydrographic partition relates to spatial differences. But my main point is it might be really interesting if you're trying to pick the right spatial domain to average a model from, to make that correspond to one of the useful spatial or hydrographic partitionings that we're already working with.
Well, we've also got like the UCSC data assimilation analysis products. We've got OI SST from NOAA. So we've done a lot of different things. This isn't the only thing we've done, but we're just generally finding that the amplitude is low, phasing isn't particularly good in the physics, and likewise gets worse for the biology. What's this one is?
This one is a model with the NOAA.
Oh, that's the OI, yeah. So
over CCS.
So there it doesn't look quite as bad on a bigger average over the whole CCS.
A lot of the SST skill comes from the forcing.
Correct.
Any other comments or questions from those afar?
Maddie has a question.
Oh, well, I have a comment. It seems to me that for physical oceanographers, satellite ocean color ended in 2010 on seaweed. I want to remind you that there is no such ocean color chlorophyll from after that, but even better chlorophyll after 2010, up to 2008 seaweed. And if you are interested in California current, then I have optimized and merged products that are combining data from different satellites and improves coverage and less gaps, and so there is more chlorophyll after 2010.
Yeah, I saw your website.
Okay, very good. I should have asked this in advance, but would the speakers mind if we post this on the website? And we've done a GoToMeeting recording. Do you have any concerns? Because part of the purpose here is to stimulate people's thinking, and it may be quite useful to have a chance to return and look at the slides and think further about them. Are the remote participants all right with that?
Sure, this is Jessica, that's fine. I can also send you my slides if you would like.
Oh, okay, well, let me consult with James. We'll see what the quality of the recording is, the GoToMeeting recording, and if needed, we'll come back to you.
Okay.
Good, okay, well, I think this is a beginning more than some concluding phenomenon, and I think there are lots of tools available that we could use to better advantage. Thank you all.
Thank you for organizing.
Great.
You should use your old joke like in class. Pretty. This is so